The maintaining of oocyte potential viability after long-term storage represents a tool of great opportunity, as it would improve domestic animal breeding by genetic selection programs, contribute to preserve biodiversity through wildlife species conservation, and increase research in biotechnology. Juvenile oocyte vitrification would also allow shortening the generation interval in breeding programs. Vitrification by ultra-rapid cooling and warming is considered a standard approach in cattle oocyte cryopreservation. However, in sheep, this procedure is still considered relatively new, and a standard method is lacking. In this video, we describe a protocol for the vitrification of sheep oocyte collected from both juvenile and adult donors and in vitro matured prior to cryopreservation. The protocol includes all the procedures from in vitro maturation, vitrification, warming, and post-warming culture.
After recovering, the cumulus oocytes complex were matured in vitro for 24 hours in starter condition. That include, for juvenile oocyte, the supplementation with 100 micromolar of trehalose in maturation medium. Following in vitro maturation, the oocytes were denuded of cumulus cells mechanically by gently pipetting and examined under stereo microscope with 60-time magnification. The selection of the oocytes to submit at the vitrification procedures represent the first important step to guarantee the successful application of this procedure. Only those with the uniform cytoplasm with homogeneous distribution of lipid droplets, integrity of the olemma, wide and continuous perivitellin space, compact zona pellucida, the outer diameter of about 19 micrometers and showing the extrusion of the first polar body, and thus at the M2 stage, must be selected. Vitrification was performed using the minimum essential volume method, with the cryotope as device. After selection, a group of 5 juvenile oocytes were conditioned at 38.5 Celsius degrees in base medium for 2 minutes. The oocytes were then dehydrated by 3 minutes exposure to equilibration solution. The loading of the oocyte in the vitrification device used is an important and critical step. Cryotope uses a polypropylene strip attached to a holder. In this method, the oocytes in the vitrification solution, less than 0.0 milliliter, are rapidly loaded with a glass capillary on top of the film strip. Then the solution must be removed, leaving behind a thin layer enough to cover the cells to be cryopreserved. Before being loaded in cryotope device and directly plunged into liquid nitrogen within 30 seconds. For warming to a biological temperature, the content of each vitrification device was transferred from liquid nitrogen into 200 microliter drops of decreasing trehalose solution. Oocytes were transferred stepwise, dragging the oocytes with a thin glass pipette to promote the removal of intracellular cryoprotectants. Finally, the oocytes were equilibrated in a base medium in incubator in 5% carbon dioxide. Immediately after warming and for each time point of post-warming culture, oocytes were morphologically evaluated using an inverted microscope with 100-time magnification. The cryotolerance of oocyte from juvenile donors is lower compared to adult ones, with lower membrane integrity after warming. The use of trehalose in maturation medium induced in juvenile oocytes higher membrane integrity, increasing the survival rates after vitrification. However, cleavage, fertilization and developmental rates of juvenile oocytes were not increased by trehalose supplementation. The use of media with calcium concentration equal to 2.2 mg deciliter for the vitrification of juvenile oocytes showed higher fertilization rates compared to oocytes vitrified with high calcium concentration, but no differences were found in embryo production. By comparing post-warming culture of different durations, we showed that after 4 hours of culture oocyte collected from adult ewes are able to recover the energetic balance and microtubular setup and to restore the developmental competence with higher cleavage and blastocytes waste. Mitochondrial activity was higher in vitrified warm juvenile oocytes after 4 hours of post-warming culture compared to other time points. Mitochondrial distribution pattern also changed during 6 hours of post-warming culture, Moreover, reactive oxygen species intracellular levels were significantly lower in juvenile oocytes at 2 hours of post-warming culture compared to 0, 4 and 6 hours. However, and in contrast with what found in adult oocytes, the rates of spontaneous pathogenetic activation increased during post-warming culture in juvenile oocytes.
One of the main advantages of the proposed method is that it includes all the steps, from oocyte collection to in vitro maturation, vitrification and warming. Moreover, it includes a post-warming culture period to allow oocyte recovery from the damages incurred during the vitrification procedure. Choosing the optimum time for fertilization is challenging, and it may impact on the outcome of the vitrification program. It should also be considered that, unlike slow freezing, vitrification is a manual technique, and it is thus operator-dependent. For this reason, a major challenge is the need for skilled technicians, and researchers should take into consideration the operator effect in the evaluation of the outcome of the vitrification program. Further studies from our lab will allow in attempting to standardize the oocyte vitrification procedure and selection, and in better tailoring media composition to the needs of the juvenile oocyte. At this regard, both the use of calcium chelator and antioxidant may offer promising opportunities.